Hello and welcome to the Brain and Transcranial Photobiomodulation Virtual Summit. I'm Dr. Joe DeJoe, your host, and my guest is Dr. Brian Bicknell. He's at the Australian Catholic University in Sydney, Australia. He's the Department Faculty of Health Science and visiting research fellow at the Photomolecular Research Laboratory. He's also the founder and director of SIMVEX. He has got his PhD from North-South Wales as a marine biologist. And he's a micro and molecular biologist by trade, not a clinician. And for his, most notably, what we're going to talk about as his experience as an outsider looking at photobiomodulation. And he's the first researcher to publish articles in science related to the interaction of light energy and the gut microbiome. Thank you, Brian, for attending and being able to participate.
A pleasure.
So tell me how you go from being a marine biologist and looking at, I guess, amoeba and things and plankton and algae or whatever you guys do, and microbiologist is at the microscope, to looking at the interaction of light and the gut. How take me through that step, if you would.
Well, I have to blame my wife for that. She has been looking at or working with photobiomodulation for 20 plus years, and I've kind of been on the periphery and watching with interest. When she finished her PhD and began her lab a few years ago, I decided to leave my teaching job and join that lab with her and help her. And then the microbiome part came with just a brainstorming session about why the remote treatment of mice with Parkinson's would help with their symptoms. And so there's a number of opinions, and because I'm a microbiologist, I said, of course, it's the microbes, it's the microbiome. So we started with a small fishing expedition, little experiment, which proved to be quite positive, and it's gone on from there.
Well, I'm excited to hear the update. I was excited about that because poop is king. We all want to know that. There's 9 times the cellular material of the bacteria than there are human cells. I mean, I've been touting that for a while, and to hear more about the interactions and how this works is a great thing, because you are sort of outside of the transcranial world, because there's a big argument that the light won't penetrate the skull, and there's a big fight that way. But can light get to the human gut? It's got layers of skin and fat and obesity. And I mean, is it really a viable option?
Well, I mean, I don't think that light is actually going to get into the gut, into where the bacteria are. I don't think that's going to be. Certainly with our mice experiments, we can expect that the light will get through. But with humans, no. I think with humans, what we're doing is we're targeting abdominal fat, and then we're setting up some chain reactions that are eventually going to change the microbiome.
Very exciting, very exciting. So we know you're in Sydney also. Would you think you're ready to go with your slides?
Absolutely. So that's not the title that you gave me, so you'll need to change that as well. But I'll email all of that to you. I'm assuming this is not going to go on the recording.
It is now. Go ahead.
Okay, what I'm going to talk to you about today is how we can treat the brain by treating the gut. So we know that it's difficult to get enough light into the brain to make changes, although we know that also works. But another way of affecting the brain is to target remotely and make changes, and that can directly affect the brain. And we know there's been a lot of research in the past few years that the microbiome is incredibly important in affecting the brain. So the microbiome in the last 20 years or so has undergone a huge increase in the amount of knowledge, and that's mostly because of much newer and cheaper technologies. Next generation sequencing can sequence entire genomes at a fraction of the cost that was possible 20 years ago, or even 10 years ago. And there are new microbiomic technologies to measure the effects of the microbiome as well. And that's led to a big increase. And almost any disease that you can think of has a microbiome component. So the microbiome is known to maintain homeostasis in many organs, and that includes the brain. It has a profound effect on our health. And we know that the dysregulation of the microbiome leads to an imbalance and leads to a lot of pathologies. I have to say a lot of that work is done with animal studies. So, and the major type of study is germ-free mouse studies. So we take mice that are born without a microbiome, we see what effects that has, and then we introduce different microbiomes to those mice and see what the effect is. There are fewer clinical trials, although there are many clinical trials undergoing at the moment. And part of those clinical trials is fecal transplants, where we take the microbiome from a supposedly healthy person and transplant into an unhealthy person and look to see what the changes are. I have to say that's in its infancy at the moment, but there are many trials, as I said. So what is the microbiome? Probably everybody has come across the microbiome in some form by now. It's the sum of the collective genome of the microbes living in and on our bodies. It's important to think about the microbiome as the genome, not the individual bacteria. And I'll talk about that in a minute. We find these bacteria everywhere in our bodies, on our bodies. Most of those bacteria are actually found in the gut, in the large intestine, some in the smaller intestine as well. There's an oral microbiome, a skin microbiome, a respiratory microbiome, urogenital microbiome, and so on. And some people even think there's a microbiome in areas that were previously considered sterile, like the lungs and the urinary tract, deep in the urinary tract, and possibly the blood and possibly the brain. There are some papers that talk about a brain microbiome. I'm not sure I actually believe that yet. There might be microbial products there, but I'm not sure they're actually microbes. Speaking about the gut specifically, there are thousands of species, maybe 1,000 species, but many, many different types of strains. So that includes the bacteria, the archaea, also protists, single-celled, what used to be called protozoa, viruses, and so on. Mostly what we know about are the bacteria, although there's more and more being learned about the viruses that live in our gut. The thing about the microbiome is most of those microbes haven't been cultured. If you can't culture them, it's very difficult to know what they actually do. A lot of them have an unknown function. We don't really know what they're doing. We can infer their function by how closely they're related to other bacteria that we know. And also we can do metagenomics, which looks at the individual genes and try and infer what these bacteria are doing. There are multiple numbers. There's trillions and trillions of these bacteria. They outnumber our human cells, depending on how you count bacteria, by maybe 10 to 1. So there's more bacteria in our bodies than there are human cells. And most importantly, they have many, many more genes, maybe 150 times the number of genes that we have. And that's why we talk about the microbiome rather than the microbiota. So the genes is important because the genes is what the bacteria do. So they have a greater range of abilities than our cells do, and that's very important. So they're a major contributor to our body's metabolomics, so the secondary products that the microbes produce that find their way into our body reacts to those, and mostly that, well, there are good and bad effects of that. If our body is in balance, our microbiome is in balance, then it maintains or helps maintain our homeostasis. And these are mostly small molecules that make it through the gut wall and into the blood eventually, or into the nerves. So we talk about the holobiome as the interaction between the genomics of our body, so that includes our cells and our mitochondrial genome, as well as the microbiology, so the microbiota genome. So the holobiome is our genes and the microbiome genes. And the holobiont is the sum of our metabolites and our microbial partner metabolites. So they're important concepts in our way of thinking. We're thinking that we're affecting the genes and the metabolites of both our cells, our mitochondria, which are actually bacteria as well, and our microbiome. There are many, many interactions. One of those interactions that the microbiome has with our gut is to absorb nutrients. So things that food that makes it through our small intestine is not digested by us, but is a food source for the microbiome. So they provide, by breaking down that food, which is mostly fibres, soluble fibres, fermentable carbohydrates, they produce nutrients for our system. So their byproducts actually feed our cells. I'll just go down to, no, I won't go down to there. There is a direct interaction with our central nervous system via the enteric nervous system and the vagus nerve. So there is a direct communication from our brain to our gut and from our gut back to our brain. It's a two-way system. And these microbes produce lots of metabolites. As I said, 150 times as many genes, lots of potential to produce lots of metabolites. They produce metabolites that are identical to what we produce, so GABA, dopamine, acetylcholine they produce. They produce small molecules that mimic our metabolites. So there's a mimic of alpha MSH, and there's lots of mimics of tryptophan breakdown from the tryptophan pathway. Importantly, they produce short-chain fatty acids. So this SCFA is a short-chain fatty acids like butyrate, acetate, propionate. That's really important in feeding the cells that line our gastrointestinal tract. That's their main food source. And so if our microbiome is in balance, we're producing lots of short-chain fatty acids, then our gut is happy. The lining of our gut is happy. Also lactic acid bacteria produce lots of lactic acid. Ammonium is a byproduct. That's not a good byproduct. And TMAO is trimethyl amine oxide, which is a bad product. It's implicated in heart disease. So as well as potentially good products that are good for our metabolome, they're also bad products. So we need to keep our microbiome in balance to keep the amount of bad micro products to a minimum. They also can produce endotoxins and whole cells, which cause a very big inflammatory response, so that's a bad reaction. And they produce ROS, and in small amounts ROS, as we know, is good, and in large amounts ROS is a bad thing to produce or have lots of.
So Brian, I think, can I just ask one question on this, because there was a lot of material there. So it makes sense that the bacteria of our gut is what takes the food and turns it into usable products for us to build a body. So it takes a hamburger and turns it into skin, right?
A microbiome?
Right?
No, mostly that's done in the small intestine, so that's not part of the microbiome. What the microbiome does is take what's left over from the hamburger, which is probably not very much, but any fibres that make it all the way through to large intestine, that's the food for the microbiome. So the more fibres you're eating, the more food that microbiome has. Also, any fat or sugar that makes it all the way through the small intestine, that will be feeding the microbiome as well. So if you're overeating fats or overeating sugars and they make it all the way through the small intestine without being absorbed, that feeds the microbiome, and that's generally a bad thing because those bacteria are not producing the right products or the good products that we need.
So what I'm asking is, the extraction of nutrients to create, good products is well studied in the small intestine, but not so well studied in the large intestine.
That is true.
That's kind of what I was trying to get to that point. Thank you for the clarification.
Not a worry. Okay, so this is a little diagram that we had in one of our papers talking about the different metabolites that the bacteria produce, and you can see there, or maybe not so well, but there's short-chain fatty acids which are feeding these cells and also producing a nice thick mucus layer. Also tryptophan byproducts, plant polyphenols, all of these are good products and ROS that will make it into or through the large intestine wall and will interact with our body's metabolome. In gut dysbiosis, where you have a bad microbiome, you have less short-chain fatty acids. It doesn't feed these bacteria, these endocrine cells so well. The mucus layer is much contracted, and you get a lot of products, microbial products, going through the intestinal wall and setting up inflammation. So a small amount of inflammation here is probably a good thing. It stimulates your immune system, and this microbiome is a really big source of potential inflammation. So it's a way that our body recognizes the outside and becomes used to it. If you set up a large amount of inflammation in this dysbiotic state, you're setting up systemic inflammation, and that leads to many things that are bad. So it leads to a lot of pathologies. We talked about already trimethylamine oxide and other products that are potentially very harmful and lead to different pathologies.
Metabolic toxemia.
Exactly. And systemic inflammation, I think, is a really important part of the disease process. It changes with your diet, it changes with stress, it changes with age, probably it changes with coronavirus as well. Okay, so what do we know about the microbiome? We know that, as I said, most of these studies have been done in animal models and germ-free models. We know that the diversity is important. So the more diverse your microbiome is, the healthier you tend to be. And we think of that as alpha diversity, which is the richness of the number of species that are there. So the more species you have, the better. Generally in disease states, the diversity decreases. The balance is important. So the different types of bacteria that are there, they're important. There are, as I've said down the bottom here, good bacteria that are generally recognized as being in a healthy microbiome. And then there are bad bacteria that are generally recognized as being in disease states or in a dysbiotic microbiome. So the balance of these good and bad bacteria is important. We really don't know enough about what makes a good bacteria and what makes a bad bacteria yet. Some of these bacteria, as I'll talk about later, Akkermansia is generally recognized as a good bacteria, but it's elevated in Parkinson's disease. So in some literature, it's seen as a bad bacteria. So the balance is really important, not just the individual bacteria. Another thing that people have looked at is the Firmicutes to...
The yin and the yang of the belly, right?
Absolutely.
Probably that thing that we're all trying to figure out there. So it even exists in there. Go ahead.
Yeah, and from that, what you don't do, I think, is take a probiotic that's one particular type of bacteria and expect that's going to fix all your ills. Okay, the other thing that's here is the Firmicutes to Bacteroidetes ratio. Some people have said that that's important in health and disease. So the smaller that ratio is, the more Bacteroides you have, the better your microbiome. Not everybody agrees with that, but it's a thing that's been published, and a lot of people think about that. The other thing that people think about is the ecotypes, the things that the bacteria are doing. So if they're making short-chain fatty acids, if they're breaking down particular soluble fibers, that puts them in a particular ecotype. Okay, so as I said, it has a profound effect on our health and also on our brain health. It's related to multiple diseases, so inflammatory, metabolic disease, obesity, metabolic syndrome, diabetes, cardiovascular disease. There's a particular bacteria that's actually an oral bacteria that's found in plaque, and is implicated in cardiovascular disease. Also trimethylamine oxide. I've got the A and the O around the wrong way. It's implicated in neurological and degenerative diseases, all sorts of inflammatory disease, lung health, sarcopenia. So it's associated with multiple diseases. So we really want to keep our microbiome happy and healthy. And for the brain, there is a dynamic connection between the gut and the brain. It's a two-way connection. It's via the vagus nerve. That's one connection. It modulates behavior. It's known to change behavior, the microbiome. It's associated with multiple diseases and disabilities. There are three potential mechanisms. One is by neuronal messaging. There's a direct connection from the enteric nervous system to the CNS by the vagus nerve. And we can, if we sever the vagus nerve, we can see the changes that occurs when the messages from the microbiome don't reach the brain. I talked about the metabolites and hormones that are secreted by the microbiome that are taken up by the enteric cells and also taken up by the nervous system, so endocrine messaging. There's also an inflammatory message process where cytokines can circulate as a consequence of the immune system interacting with these microbial products. So there are three different mechanisms where you can get interaction between the gut and the brain. I'll just talk a little bit about how the microbiome interacts with the brain. So it interacts in neurodevelopment. There's not very many longitudinal studies, most of they're cross-sectional studies, but we can see that microbial or microbiome diversity does affect development. It affects neurogenesis, it affects myelination. So germ-free mice don't develop as well as mice with a normal microbiome, and mice with an altered microbiome, a humanized microbiome, don't develop as well. And there's also this correlation between alpha diversity, so the richness of the species, and cognition. So if you are microbiome deficient or poor in your early life, that is reflected a number of years later in cognition, and that's in human trials, small numbers of human trials. In neurological disorders, the microbiome is heavily influencing neuroinflammation, inflammation and neuroinflammation. In this case, it's probably small scale changes, so individual bacteria rather than overall diversity changes. So some key beneficial bacteria have been identified, and that's the basis of supplementing with probiotics or changing the microbiome for particular disorders. For example, autism spectrum disorder. There are some bacteria that are thought to increase or decrease the symptoms of autism. Kids with autism spectrum disorder tend to have a lower diversity, but it tends to be correlated with particular types of bacteria. They tend to have constipation, they tend to have a low fibre diet, and so the symptoms can be changed and have been shown to be changed by fecal transplants and by dietary changes. It's more difficult to change things like Alzheimer's disease. People have tried diet, they've tried changing probiotics, changing prebiotics, but a very difficult thing to change or to slow down. There is a very strong vagus nerve connection with Alzheimer's, and that's because the bacteria, the dysbiotic bacteria in the gut, can produce an amyloid protein, and that can be transferred via the vagus nerve and may set up the increase in amyloid protein that's characteristic of Alzheimer's disease, so the beta amyloid and the tau plaques. Epilepsy, again, germ-free mouse studies have shown a link with the microbiome and epilepsy, and the fact that a ketogenic diet can alter epilepsy is strongly suggestive of a microbiome involvement. There's less evidence for ALS and Huntington's disease. There is some preliminary evidence. For Parkinson's, there is a very strong gut-brain link by the vagus nerve connection. And if you listen to Anne Leavitt's talk a couple of days ago, she would have said that a vagotomy for treatment of peptic ulcers in times past actually is protective against getting Parkinson's disease. So severing the vagus nerve, and this is also in animal models, if you sever the vagus nerve, the animals won't get Parkinson's. Parkinson's often begins in the stomach, in the large intestine, so gastric symptoms. Often Parkinson's patients will have diarrhea, uncontrollable diarrhea, or constipation, so major dysbiotic gut microbiome. The microbiome also, some of the microbiome bacteria produce dopamine. So dopamine production it's not sure how important that is, but there are bacteria that do produce dopamine. There are also bacteria that break down dopamine. So the L-dopa that patients take, some of that is broken down in the gut, and so it doesn't actually make it to the brain. Even though there's been a lot of work done, there's no consensus on a microbial signature, so we don't know what bacteria are indicative of Parkinson's. We know that there are some bacteria that are more common in Parkinson's, and there are some bacteria that are less common in Parkinson's. There's been a recent study in Finland, an epidemiological study, where they've shown that taking antibiotics of particular types will increase your risk of Parkinson's. So an antibiotic is a bad drug to take for your microbiome. It'll kill a lot of bacteria. That is related to Parkinson's. On the other hand, if we could work out which bacteria were the signature of Parkinson's, then perhaps we could treat Parkinson's with antibiotics. There are also some fecal transplant and diet trials going on with Parkinson's. They may help with constipation. They don't seem to be great at reducing other symptoms of Parkinson's. There's a strong connection with the microbiome and migraine, and consequently migraine and Parkinson's disease, and also schizophrenia. I'll just skip through these now. There are probiotic studies for schizophrenia, and we can change some of the symptoms of schizophrenia, and behaviour. We can change, mostly in animal models, we can change behaviour by changing diet, by changing the microbiome, by adding probiotics to the diet. It's a good study where they showed that anxiety in mice was, when they're going through a very complicated maze, anxiety was reduced by feeding them yogurt. Okay, we can change the microbiome, as I alluded to. We can change the microbiome with diet. That's probably the most profound way of changing the microbiome. There's a distinct microbiome for a carnivorous diet versus a vegan or plant-based diet. If you increase your consumption of fat and sugar, that increases or changes the microbiome, and not in a good way. If you increase the soluble dietary fibre or the fermentable carbohydrates, that also changes the microbiome. So because these bacteria are eating the food that we eat, what makes it down to the large intestine, that's the obvious way to change the microbiome. It's actually quite easy to change the microbiome in a bad way. It takes longer to change the microbiome back to a balanced microbiome. Stress will change the microbiome. Aging will change the microbiome. Microbiome changes with age. Diversity decreases. And that's correlated with health outcomes. So as you age, if you can maintain the diversity of bacteria, then your chance of inflammatory type diseases decreases. We can also change it with drugs. Antibiotics, that's a negative way, mostly a negative way to change your microbiome. It's going to be very indiscriminate in the bacteria that it kills. In our studies, if people have been taking antibiotics in the previous 6 months, they're excluded from the study because it takes a long time for the microbiome to get back to something that's like a balance. And then there are the anti-diabetic drugs, metformin, TZDs, and GLP receptor antagonists. They change the microbiome in a positive way. So one of the ways that these anti-diabetic drugs work is by changing the microbiome. We can change by prebiotics, which is taking dietary fibre supplements, or probiotics, which is eating the bacteria themselves. And as I said, it probably needs to be quite a big range of bacteria. Or postbiotics, where we're taking the products that the bacteria are producing, or we're eating dead bacteria, so we're eating their metabolic products. All of those can change the microbiome. Fecal transplants have been tried quite a lot. Clostridium difficile infection, that's quite good. There are trials for irritable bowel syndrome, Parkinson's disease, depression, mixed results so far. What we're interested in is can the light affect the microbiome? We know that circadian rhythms affect the microbiome. If you're jet lagged, that affects your microbiome. The microbiome itself has its own circadian rhythm that's in concert with when we eat, with our liver's circadian rhythm and our body's circadian rhythm. And vitamin D directly affected by light, which also the vitamin D directly affects the microbiome. But what we want to know is, can PBM change the microbiome? Can light specifically targeted change the microbiome? As I said, there's been many experiments with diet, with pre and probiotics, with fecal transplant, with drugs. Less than ideal results so far, but probably early days. And there are many clinical trials going on. It's more difficult with clinical trials because you can't, there are multiple factors involved in clinical trials that are hard to control when you're doing a microbiome study. Okay, so why microbiome? Why PBM for the microbiome? We know that there's a systemic effect of PBM, and decades of work at Sydney University on Parkinson's and also Alzheimer's models of disease, Parkinson's and Alzheimer's models in mouse and monkeys, using toxins and using transgenic models. They've shown that you can decrease the symptoms of Parkinson's and actually precondition against Parkinson's using both transcranial, intracranial and remote PBM. And there was one experiment that we rely on, which is called the helmet experiment, where they shielded the head from light and just shone light on the abdomen, and that was also protective against Parkinson's. So we had a brainstorming session about why this would be, and myself as a microbiologist, I said, of course, it's the microbiome. What else could it be? So we did a small experiment, a small fishing expedition to see if we could show that the light changed the microbiome. And other people are thinking about this as well. There's a group in France that are also thinking about Alzheimer's and the microbiome. And we know that there's a strong gut-brain connection with Parkinson's disease. And what I'm saying now is basically based on 2 papers that we've published, but also I'll give you some unpublished results from our Parkinson's trial as well. So the experiment that we did was very simple. We had 4 healthy mice per group, 5 different groups. We had a single dose of red light, multiple dose of red light, single dose of infrared, multiple infrared, and then a sham group. We took feces at days 0 before treatment, 7 and 14, so not very many samples. We extracted the DNA, we went through a next generation sequencing process, and we analysed the data. So a fairly simple experiment, took about 6 months. We analysed about 10 million sequences. We got rid of the low quality sequences. We found about 2,900 operational taxonomic units or strains. We found no archaea, so no methanogens. We saw that there was no significant difference in alpha diversity between the groups, so that the diversity didn't change between the groups. The richness of the species didn't change within the groups. But there was a significant difference in the beta diversity, which is the variation between the groups. So, thank you for that. Okay, there's our p-values, but if I go there, that shows you where the significance were. So there was no difference between light and a single dose of red, but there was a difference between no light and multiple doses of red. So you needed multiple doses of red to change the microbiome significantly. But a single dose of infrared or a multiple dose of infrared also changed the microbiome. So we've seen no other changes, no diet changes, mice in the same cage, having a fairly similar microbiome to begin with. The only thing that's changed is the amount of light that we've given them, and we can see a change in the microbiome. Graphically, the red dots are the no light, and you can see that they cluster a little bit separately to the ones that have had a light treatment. So in that that principal component analysis plots. Okay, they're the genera that we found. We said that all the mice were fairly similar, but you can see their microbiome is a little bit different. Down the bottom here, we've got the multiple red light, multiple infrared, none, which is no light, which includes both the sham and the time zeros, where that's before they had any light treatment. And then we've got the single red light and the multiple red light. I think that's right. No, single red light, single infrared. So what we need to do is find out where that, we know there is a difference between the multiple and the none. What we need to do is find out what that difference is. Okay, these are the bacteria that we found, the strains of bacteria that we found. So we need to look into that to find out where that change has occurred. There was one bacteria that was statistically different between those groups, and that bacterium was called Alabaculum. That's a bacteria that's found in mice and also dogs and rats, but not in humans. So it's not really relevant to people, but it is to the mice. And whereabouts are those bacteria? That's them down there. So you can see that green bar down there, that's the change. And you can't actually see it in any of the other groups. So the change was driven by a fairly small change in one species, or statistically speaking anyway. This is a graph of the numbers. You can see that the numbers by day 14, the numbers in the multiple 808, multiple infrared, had increased 1,000 or 10,000 fold over what you saw in day 0. So a major increase in numbers of this bacterium. And that is a good bacterium. It's beneficial in obesity and prebiotic treatment. It's a short-chain fatty acid producer. It's implicated in a healthy mucosa. Interestingly, it's also increased by metformin. So if you did the same experiment using metformin, you'd also see the same type of increase. And it's increased with fibre and decreased in obese and high-fat-fed mice. So it's a good bacteria. We've increased it by using light. But remember, we've started off with what are apparently healthy mice. We haven't changed very much, but we've changed enough to show up statistically. We've changed this good bacteria for the better. Okay, that was a very small experiment. We've done this a number of times now with other mice, bigger numbers, multiple treatments. In the end, we find that infrared is still better than red. That may be a penetration issue. And we find increases in a number of bacteria, and they're all good bacteria. We've also found decreases in some bacteria that are thought to be bad bacteria in mice. So we've confirmed it in mice. Does it mean anything in people? The literature is full of things that work in mice that don't have any application to people. Okay, so yes, we've confirmed it. We've seen after 14 days, so it's most probably a secondary effect. We're probably not affecting the microbiome directly. The generation time of these bacteria is something like 40 minutes, so you would expect to see large increases in a very short time if we were directly affecting the microbiome directly. So we're probably affecting the body, which is in turn affecting the microbiome. As I said, in people, do we know if it's important or not? There's only a 30% similarity in our microbiome to mice. Why do we use mice as a model for people? It's a really bad model for microbiome. Okay, but this does have implications for our Parkinson's treatment. So we use this as a basis for deciding what outcome measures to use in our Parkinson's trials. Before we did that, we had a few unpublished human studies, and we saw changes. You can see the the change in beta diversity between the groups is quite dramatic. This particular person was a pre-metabolic person. Change in Akkermansia, which we said was a good bacteria, from non-detect up to about 20% of the total microbiome. So a major change in Akkermansia. Also changes in these other good bacterium, Bifidobacterium, Bacillobacterium, Rosiburia, and decreases in these so-called bad bacteria, dysbiotic bacteria.
Oh,
So we have confirmed it in a number of human studies. Yes.
so my question here is that the alpha diversity didn't change that much, which is the total difference.
Total richness, yeah.
Total richness. So the whole balloon grew, but you saw a difference in the beta, which was the good to the bad. So is that
the treatment to the pre-treatment?
The treatment to the pre-treatment changed.
Yep.
The good,
but the overall richness didn't change.
But the overall, so so you had to have more, but you did have more good. So wouldn't that make it more diverse, though, if you had more good than bad?
Not necessarily. You still got the bad bacteria there as well. You haven't got rid of them completely. Uh-huh. And what you're doing is really changing the profile of the diversity. So you're changing the balance, increasing the good bacteria, decreasing the bad bacteria, but the overall diversity hasn't changed.
Well, so like there was no immigrants.
Statistically speaking, no new immigrants. That's right.
All right, there we go.
Okay, so we're calling this photobiomics, where we're using light to change the microbiome as well as the whole of the holobiont, the cells, the mitochondria, and the microbiome. Okay, so we translated this to our study in Adelaide and in Sydney for our Parkinson's trials. For the details of this, you'll need to go back and listen to Anne Liebert's talk again, but in simplistic terms, we had 2 groups in Adelaide, 6 participants per group. The participants were self-selected. They answered ads to go on the trial. In Adelaide, we had transcranial, intranasal, abdominal and neck treatment. We treated for 12 weeks and we titrated the dose down. So 3 times a week for 4 weeks, then twice a week, and once a week, and then they went onto a home treatment. In Sydney, we didn't titrate the dose down, and we actually doubled the abdominal dose. So the abdominal dose in Sydney is about 90 joules, and in Adelaide was about 40 joules. So quite different treatment regimes. In Sydney, only abdominal. In Adelaide, transcranial and abdominal. That's our treatment regime. We used the V-Light device transcranially. We used a laser on the neck and then on the abdomen. We had no change in diet or exercise as far as we knew. We asked people to keep diaries of their exercise and diaries, well, not diaries of their diet, but to talk about their diet and what had changed. We tried to make sure that there was no dietary change. Very difficult to enforce that over a 12-week period. We used, as I said, laser to the abdominal and neck in Sydney, plus transcranial in Adelaide, and we took multiple samples. What I'll talk about is the before treatment and the 12-week of treatment microbiome, because it gets very complicated looking at all of those weeks. Okay. So we're looking at beginning of before treatment and then at the conclusion of the trial. We had about 170 million sequences that we looked at. We saw no statistical difference between the before and after treatment for either alpha or beta diversity for either trial. This is not surprising because they're small numbers, and men are not microbes, or sorry, men are not mice, because we started with our mice. They all had fairly similar microbiomes. With people, the microbiome is very, very different. So with only 19 people, the beginning baseline is very different between. And so to see a difference between the beginning and the end of treatment, you would need many, many more samples. So no difference in either alpha or beta diversity for either trial. But if we combine the two trials, then we did get significant changes in alpha and beta diversity. Okay, so if we combine the two trials, there is a difference between total richness from before treatment to after treatment. There's also a difference between before, in beta diversity, the makeup of the microbiome before to after. Now, I would never do this in a publication because the treatment regimes were different. But it gives us a good handle on the fact that we are changing the microbiome. Overall, there was a decrease in the Firmicutes to Bacteroidetes ratio in 15 of our 19 participants. And remember, I said that's maybe an indicator of health in the microbiome. So it suggests a healthier microbiome in 15 of the 19. And generally, there's an increase in the good bacteria, what we say the good bacteria, and a decrease in the bad bacteria. But again, it's highly individual. I will go quickly through some of these results. These are still preliminary. They're unpublished. The actual numbers will probably change a bit before we publish. That's like a heat map of the changes. These are the healthy bacteria. The blue, there's 3 shades of blue there. The blue is increase. The red, the 3 shades of red, is a decrease. So you can see there's more blue than there is red in the healthy bacteria. There's more red than there is blue in the unhealthy dysbiotic bacteria. This one, indeterminate bacteria. So we don't know. There are contradictory results about whether they're good or bad in different publications. So this is what we're looking at. We're looking at how many of these participants showed an increase in good bacteria and how many showed a decrease in the bad bacteria. So I'll just quickly go through those. Ones that are recognised as good bacteria generally increased. And by increased, I mean by a factor of 2 logs, 2 log to the base 2s. So we see a lot of variability, but we're saying you have to have 2 log 2 changes to be significant. Okay, these bacteria are all recognised as good bacteria, healthy bacteria. 8 participants showed an increase, 2 showed a decrease, 6 showed an increase, 1 showed a decrease, 13 showed an increase, and so on. These bacteria down here, there are mixed reports. This bacteria sometimes is shown to be increased in obesity, sometimes it's shown to be decreased in obesity, increased in leanness. And we found 9 of our participants showed an increase, 2 showed a decrease. Fusobacterium, there's one particular species of this that causes or is very highly associated with colorectal cancer. Not this particular species, I have to say. We showed an increase in 8 participants, a decrease in 1 participant. These are bacteria that are known to be or thought to be good, and they're reported as being lower in Parkinson's patients. So they're possibly a marker for Parkinson's. We talked about that one before. Fusobacterium, Roseburia, Prevotella, all increased in the majority of patients. Oh, I've got Prevotella twice. That one should be Para-Prevotella, increased, decreased. Then we have some good bacteria that are recognised as being good, but for some reason are elevated in Parkinson's patients. So there's a disparity there. And I talked about Akkermansia before. It's increased in leanness, it's increased in good health, but it's also increased in Parkinson's. And we found that it increased further in our Parkinson's patients, and also Parabacteroides, a good bacteria but also increased in Parkinson's. We found it increased further. Our bad bacteria generally decreased, again the same 2 log change. Diarrhea went down in 13 participants. Blautia went down in 9, and so on. Curibacter went down in 11. This one, that family went down. And these bacteria are reported as being high, so potential marker bacteria. And we found most of these went down in the majority of our participants.
Wow.
Bifidobacterium is interesting because sometimes it's a good bacteria, sometimes it's a bad bacteria. It depends on the species. At the moment, I've only gone to genus level. I need to dig down into the species level to say more about that. Sorry, you had a question?
No, I mean, I'm just surprised, and you must be ecstatic and seeing these directional changes. At least we have the needle is going in the right direction. That's very, very positive.
These ones, again, there's different reports that say that there different things are happening with these bacteria. And these ones are all going down with our Parkinson's participants, so possibly they're new potential markers for Parkinson's. So, and yes, you're right. I'm very, I won't say surprised. I like the fact that everything is going in the right direction.
That's right.
But early days yet. We have to do a much larger trial, of course. For our other outcome measures, our Parkinson's participants fell into 3 groups. Super responders, 11 of our 19 super responded to the treatment, got better in a lot of the outcome measures. What I'm doing now is correlating that, or coinciding that, with the microbiome. And it looks at this early stage that there is a good correspondence between the ones that had big changes in the microbiome and better outcome measures. We're going to have to dig a lot deeper to work out cause and effect, or what's driving what. And we have potential to look for specific markers for disease progression and for disease treatment. So the implications of that, I think, are quite profound. The microbiome influences the brain. We know this. We know that we can change the microbiome using laser, PBM. I have to say I tried LED. It didn't work as well, so we tend to be laser people. We know that the people that we've been testing already have a variable microbiome, but we can change it in a positive way. In Parkinson's disease, there hasn't been much success with changing outcomes and symptoms using diet or pre and probiotics, or even fecal transplant, mixed success with fecal transplant. We think that laser has a great potential to be a treatment for at least symptomatic relief of Parkinson's, or remote treatment on the gut to affect the microbiome and then affect the brain. We obviously need a much larger trial. We need a placebo arm as well. We didn't have any placebo arms in those first 2 trials. And we need to do a lot more work on the microbiome, but we are very, very hopeful. Thank you.
Thank you, Brian. Now, let me ask you a couple of questions to kind of lend your expertise to this. Now, you suggested at the beginning that the light is affecting the body of the person, which then is affecting the microbiome, the microbiome, right? Mm-hmm. Now, and then it's going to kind of grow, and then its diversity is going to change. The good and the bad are going to change. So do it to me, be a weatherman for me. So if you're looking at it, I tend to think of the microbiome like the rainforest. Things happen good, it rains and it changes quickly, and then things grow faster, and it's more fertile over here, and this part gets. So how is this working? If I have if I have an event and it creates more good bacterium, that's going to have a a decrease in inflammatory response that's going to go through the whole body. It's going to have a decrease in inflammatory response that's going to be reflected through the whole body. It can create the hormones and things like that we talk about that can have a calming effect or an agitated effect if you're stressed the other way. So it's sort of like it is a signaling system, though, correct? Is that what we're saying?
Absolutely.
So by having doing the right things on the inside, and then it's kind of it's a weather map or something. How do you view it? And when you look at those maps, what do you see?
I don't think of it as a weather map. I'm sorry. We think of it as it's a very positive feedback loop. So you're changing or affecting the body, that's affecting the microbiome. The microbiome then feeds back to reduce inflammation and produce those good metabolites, which is affecting the body. So it's feeding on itself. The thing with Parkinson's, though, is you need to continue the treatment. So if you stop the treatment, the symptoms get worse. We showed that in our trials. So I think that the body will, the Parkinson's disease will change the microbiome back to a bad state, as if you don't continue the treatment and continue that process. So I think it's a positive feedback while it's working. And I think if you combined it with diet and exercise as well, the effects would be even more pronounced. We told our participants not to change their diet. Interestingly, a lot of our participants felt a lot better, felt healthier, and wanted to change their diet. They wanted to feel healthier and change their diet to the better. We told them, please don't do that.
Yeah, and they always say that if you start to, they said that if an alcoholic starts drinking orange juice, it's not that the orange juice cured him; it was not drinking the alcohol.
Right.
So when you start to feel good, you want to do better and do the better things for you, and
exactly.
That is still the connection. Good things are happening, and you're feeding forward, and so I think that you're observing that also.
Yeah.
So
absolutely, and in a way, it's difficult when you're doing a trial because you want to keep everything, as homogeneous as you can. But you also want the best for the participants. You want them to, feel the best that they can and improve their symptoms. And that's a bit of a problem.
So answer me this, if you can, just to, with your theories and stuff. What is the major kick in the pants in the right direction for the microbiota, for the microbiome, when you put light on it? What is it? Is it an energy thing? What is it that kickstarts this biome?
Personally, I think it's a reduction in inflammation that's going to start kickstarting that microbiome change. We're really at the beginning of this. We really need to get a much better handle on it. But the thing with Parkinson's is that it doesn't change much if you're going to change your diet, change probiotics, change prebiotics. So whatever we're doing, we're giving a bigger kickstart than doing just a diet change.
I like that's very positive. Now you have a little incubator there, if you will, down in Australia, where you have formed a new company that's trying to do well. What is your objective? Because as I said, when we come back for the 2030, and we put our Back to the Future helmet on. And what do you see? How do you see this work participating in the future? What do you see for the future?
Well, the purpose of the startup company is to get the treatment to the patients. And I think doing it commercially is the way to do that. I think trying to go through the medical profession and try and convince them that something is happening, it's not placebo, that's a very difficult thing. We had a lot of trouble with neurologists doing this trial. We had a neurologist who began with us and then gave up, before we saw the end of the first trial. Most doctors will not give light the time of day. So we thought that the best way to get to the patients is to commercialize and, get the treatment out there.
I'm so for that. The Australia's an island, and here somebody starts to do research with light therapy is good for the brain, and next thing you know, they're not waiting for their doctor. They're just building the thing and putting it on my head and saying, I'm just going to do it. I like it. And I think that you're being able to address this cataclysmic, brain stuff that's going to be coming down the pike. And at least you guys are building some infrastructure, some cushion to be able to have a public that's aware of it and know that they have some actions to take. So I salute you. I support you. And we'll see how it goes. We'll look forward to good news as we come forward. Thank you, Brian.
Thank you very much.